Good morning, and thank you all for being brave enough or energetic enough to join us bright and early on a Saturday morning. Let's pull this back a little bit. Can you still hear me? It's not as echoey? Okay. The title of this session is called Selling Stem Cells, and we've taken a little bit of liberty with that title, and what we really want to talk about is the broad market for stem cells and stem cell research products, therapies broadly. I'm going to have my panelists introduce themselves in a minute, but let me begin by introducing myself. I'm Bob Hutchens. I'm the president and CEO of StemBioSys. We have, I would call it 1.5 products right now. Our first product in the market, and it's actually available now, is an extracellular matrix used for the expansion of cells. Most of that work that we've done to date has been in the area of mesenchymal stem cells. On Wednesday of this week, we announced a collaboration with BioBridge Global, and we will be entering the stem cell supply market in the middle of next year when we work together with BioBridge, and we'll be selling cord blood cells or mesenchymal stem cells isolated from cord blood, and we're looking forward to launching that product in June. So we're relatively new to the stem cell supply market, but as we'll talk about in a minute, there's a lot of pieces to that market, and we've been around in that space for about a year now. So let me ask my panelists to briefly introduce themselves, and then I'll make some introductory comments, and then we'll get started. So, just a brief intro.
Okay,
yeah.
Good morning, ladies and gentlemen. My name is Hirao from Cellular Dynamics International, MyCell, Wisconsin. As you know, Fujifilm purchased Cellular Dynamics International in this early May, and then I moved into Madison's in August. So then I would like to a little bit explain our companies based on the slides afterwards. But today I'm very much pleased to speak to you, and then also hopefully we'd like to have good discussions after the presentations. Thank you very much.
Thank you. Good morning, everyone. My name is Asutosh Yagnik from Aura Capital Partners. We're headquartered out of Iceland, which is, I think, probably wins the prize for the most unusual location of the companies that are here. It's not surprising, the founder and CEO, David Levinger, spent a lot of time there, and given the biotech industries where a lot of them Decode and other companies were based out of there. There's a lot of good stuff happening out there. I'm based out of London, and my background was initially in academia and then pharma, research, R&D. Went into consulting and have been doing consulting and independent leadership development for some time. Aura Capital Partners focuses, it's an independent, privately owned, management-owned company. We actually help senior managers, leadership teams in companies focusing mainly on the technology and knowledge-led industries, and we help organizations really with strategic consulting and hands-on operational management, looking at how we can help them change and transform their businesses and focus on the future. We have 4 basic areas. That's capital sourcing, strategic consulting, global communications, and transformation.
Great. We're going to do a couple of things to begin to set the stage this morning. I'm going to walk real quickly through some numbers, and then my colleague here, Kaz, is going to walk us through a little bit about the cellular dynamics story, and I think answer the really interesting question. I know when we met for the very first time yesterday to prepare for this panel, the very first question I asked him is, so what attracted Fujifilm to this market space, and Kaz has some really interesting slides that he'll walk us through in just a minute around that. But I did want to share with the group some data. I did some digging this morning in preparation for this, and we looked at a study that Bioinformat did last year. It's called Stem Cell Research Products. The study was published in June of 2014, and just data I think is really interesting to ground ourselves in this. They define the stem cell research market, the stem cell research product market globally, and this excludes antibodies. They went through an exercise of estimating that market, and in 2013 they estimated that market to be about $1.75 billion globally. That's B. So it's a big market. And interestingly enough, when they did some projections in terms of where the market's going, they estimated it at just over $2.5 billion in 2018. So you've got about a 40% growth rate occurring in a relatively short 5-year time frame. So again, there's lots of growth here. Interestingly, too, when you look at the players in the market at the time of this study, Becton Dickinson, Sigma Aldrich, and Thermo Fisher had 80% of that market. And then the tail, 20%, was really a lot of little companies that very specialized offerings. Companies like ours and Cellular Dynamics were included in that tail at the time. The other thing that strikes you, so you've got a big market that's growing. The other thing that struck me as I was looking at it is there's lots and lots of collaborations that occur in this. Even a big company like Thermo Fisher, there were 7 collaborations at the time that they had with academic institutions and other companies to prosecute various opportunities. So even a company seemingly with relatively unlimited resources by the measure of many of us still need to collaborate because the knowledge of this field is all over the place, and no one organization has the market cornered on that. Some other interesting data that as I looked at this, about half the market split pretty evenly, so about a quarter of the market is in MSCs, and they actually went and looked at publication data to see what was the research that was being published this was in 2012. Another quarter was HSC applications, and then about 40% split pretty evenly, embryonic cells, cancer cells, neural cells, iPS cells, and cord. I suspect that if we were looking at that data in 2015, we would see a different mix, but that's what it looked like in 2012. And then in terms of why people buy and what people buy, kind of interesting, the primary reasons for purchase were around cell differentiation and cell culture in terms of the kinds of products people were buying. Probably no surprise there. And then the final thought, just in terms of numbers again, I thought these were kind of interesting. They went out and looked at the number of stem cell clinical trials, and I'm sure there's been data around this all week, but again, to give you context, in 2009, they identified about 2,300 stem cell trials going on around the world. In 2013, that number was up to 4,200. And what was really surprising to me, as an American, is that 2,400 of those trials were actually here in the US. I would not have guessed that, hearing that number, that more than half of them were here in the US. And then one final statistic to think about, in terms of publications in stem cell related field, they identified about 8,300 publications in 2004, and that number grew nearly threefold to 23,000 or so by 2013. So lots and lots of growth, and as you think about this in the context of our discussion today, there's been a lot of growth to date, and certainly as we look forward, I think we're going to see lots of growth too. So with that, I'm going to turn it over to Kaz, who's going to walk us through a few slides to, number one, just tell us a little bit about cellular dynamics. But the piece, I know when I looked at these this morning, the piece that was really interesting to me is why did Fuji enter this field? What did they see and what was so attractive about it? So, yeah.
Thank you very much. Then I would like to use slides because of the second language handicap. So please see the actual slides anyway. Then first, I would like to briefly explain our companies. That means you can understand, so for the IPS space, what kind of things has been done so far. And then based on that, we would like to really explore, so why Fujifilm actually interested in this space, and also what is the current challenge for us, and maybe opportunity on the other hand. Here is a very brief summary of companies. CDI is the world's largest producer of human iPS-derived cell productions. And then, as Bob told, actually, currently MSC is a very huge market. And then iPS-related cell has not been so much any portions of the market. And then the company has founded in 2004. As you know, Dr. James Thompson was the founder of this company. Then based on that, we actually developed a lot of iPS-related technologies in this company. And then April 30th, Fujifilm bought this company. And then now around 167 employees. Then based on the around 10 years experience, we think the cumulative experience of companies is 700 years equivalent. And then we have 900 patents above. That's why, so we can provide customer with free to operations. Customer doesn't care about any patent problems in the future. And we now strategically focus on the two markets. First one is life science research market, and the other one is cellular therapeutics market. As you know, currently life science research part, iPS cell was tried to be used by big pharmas as well. And then we think this is really a launch of the market that is a phase. And the cellular therapeutic part is MSC, for instance, well, a lot of trials already getting in, but iPS part is not clearly studied clinical tests. And then we are also trying to get into that market as well. And then this is a brief, actually, product lines. We have actually around 10 iPS-derived cell products, starting from cardiomyocyte, hepatocyte, or the neurons, those kind of things. Then here, very small, but this is actually, we actually are applying ISO for the research product quality control and the GMP for the, of course, clinical control. And the GMP facility part, we have not yet actually owned by ourselves. We are now collaborating with the University of Wisconsin. Then we had a lot of actually collaborations. As Bob already stated, in this industry, we need the collaborations because we have no knowledge to produce a good cell, but in order to use cell, then we need assays or other actual technologies to be getting to the customer's usage. So then a lot of actual collaboration was done for the past, and then many papers, and then now actually maybe two remarkable things is first one is FDA approved CGMP release testing of botulinum toxin. So then FDA actually approved the standard of that toxicology test. So that is a gold standard will become based on the iPS cell. And then the other one is FDA is evaluating iCell, which is actually arrhythmia. This is now under working, but probably end of 2016 or early 2017, ICH will rewrite the guideline for this test. So then, in that case, iPS will be used regularly. So those two are, I think, a remarkable, actually, achievements so far. And then, this is actually just simple illustrations how actually we are managing qualitative or any quality cells. So starting from the actually donor samples and then establishing the iPS cell, then making a differentiated cell. And those processes are totally controlled. And then our production scale operations has been built up. That is the reasons why so billions of cells has been already produced in our company. And also a variety of cells has been developed. And then in order to sell the cell, we're focusing on several things. First one is how to make a great assay. This assay can make people do the new things. In the past, we cannot do, but iPS cell-based assays we can do. So those kind of a new value market has been created. And then we have three actually different, of course, approach, innate and induced and engineered cells. And those three we already established internally. For instance, first innate examples, this is diabetes-associated cardiovascular disease. Based on the SP and FP donors, and we actually picked up their samples and create the cardiomyocyte. And then these actually functionality has been checked. And then this is actually pretty much reflected the disease background in these three different cells. So then, based on this, by the way, this is actually Roche and the MGH were collaborated to use this assay. Then based on this one, we actually tried to screen what kind of compounds can be effective for those kind of disease. So then this is actually illustrated first starting 47 hits, and then lastly actually 18 hits has been derived based on these assays. So this is just examples. Because by IPS, so we can actually get different background disease or gene samples very easily, and then that can be actually reproduced based on the productions. So that's the reason why. So this kind of phenotypic type of screening can be very much empowered. Then this is another example. This is actually based on the induced actual model for using actually endothelin. Then you see this BNP actual level can be clearly indicated in the graph. And then also you see this Z-score is very good to be used, this one in the assays. And then this one is actually gene engineering examples. We actually, use, this one is epidepix, actually epidepicity models. And then these also graphs shows this cell can actually represent this epidepicity disease based on actually gene modified P924L part. This mutation has been created. So then that is very clearly actually have the impact on that. And then we have those assays right now. So then we think actually this can be used for the drug discovery right now. So this is the current status which we did so far. And then now, world is a little bit changing because as you know that there are many iPS cell bank is now available in the world. So that is, we think, a very great chance to make a new world of the drug discovery and screening processes, and then that will be efficient for the pharmaceutical companies. We call this as disease in a dish. That means the many disease model and the gene background different models cell can be produced. That is actually going through the screening. That means drug actually assessment can be done in vitro, not before using vivo. So that means a more efficient actually screening can be done. So that's the concept. And then this is just examples how many iPS cell bank is now emerging in the world. And then we are several actual cases we involved in these activities. And then maybe first one is the SAM. So they actually give us a grant to establish at the end 3,000 donors, different disease. This is listed the different disease, and they actually collected 11 different disease background donors. And then we create those actually donors iPS cell and then this is available for everybody over the world. So then we can actually make a jump start to have such new world of the drug discovery. This is another case. This is actually the NIH grant we got, and we actually produced 230 iPS cell for, this is actually cardiovascular disease actually background model. So then what I want to say is currently these different iPS cell is available in the market. So that means many drug companies, pharmaceutical companies would like to use those kind of cells for their drug screening. That can be change the world in the near future. We are now feeling that. Then in order to do that, most important technology is how to make a purity, high volume cell in a short time. So this is actually, we think, a very competitive edge of our company. So we have those kind of capabilities and know-hows. And then this is examples for the NIH grant case of different donors. And you see the purity of the cell is always 95% above. So this kind of technology is very important, we believe, because then assay can work properly, and then people can actually assess the efficacy, those kind of things very accurately. A little bit different topics. So we have two focal areas. So this is another focal area, therapeutic part. This is current actually our active pipeline. Still, iPS therapy case has not get into the clinical stage. The most first one is this ocular ARM this therapy part. This is we are expecting 2017 IND. Maybe first day of this session, Dr. Kapil explained the status of that. We are heavily involved for that project. This is actually the project which I said. Now actually pig study has been more or less finalized. Then our CDI actually role is establish those compatible protocols of the CGMP facilities. That means based on these protocols, this cell can be produced under the CGMP facility. And then you see actually those kind of protocols, and then at the end, a very black face RPE was produced. This is actually very high purity. This is Datus. First actually established cell is a very variety of the purity. But after purification processes, those cells actually purity is around the 98% range. So then this protocol is now going to the FDA actually assessment toward the IND. Another thing is the cardiac programs. This is still very early because still actually animal test and then very early stage, but several actually some efficacy point of the fixing the heart failure has been confirmed. And the Parkinson's disease program is next one. This is actually we producing dopaminergic neuron, and then very actually precise TAC is found, and also this one is engraftmented to the rodent, the rat, and also the currently monkey. And the rat case, you see the efficacy has been approved. Then now is monkey result is waiting. Then last thing, what we are now doing is iPSC HLA bank. You see that this phase 1, we actually now established 2 lines of HLA iPS cell, and which is around 35% US population is covered. But then you see that this kind of curve, then maybe five HLA types can be covered around 50% of populations. And then we have a very great source of the donors. So then based on the different actually people, and then probably we believe each country's maybe 50% coverage can be designed in the future. So that directions we are now working for. And then Bob's first question is, what about CDI attractive Fuji? This is a very understandable anyway, way of considerations. First, market attractiveness. Bob has explained, so very huge growth is expected. And this is a graph. Source is the Ministry of Economy, Trade and Industry in Japan. So our government, Japan government has also see stem cell actually related business very high growth. So that is the reason why Japan is very much stressing on this business. Then they actually changed regulatory last year, November. Then Fujifilm actually saw this market opportunity is really, really huge. And the next one is a strategy fit. After collapsing film businesses because of digital impact, we actually planned to diversify our companies. And one of the most important directions is healthcare. Now healthcare sales is around 18% of total company revenue. Then in order to actually make healthcare businesses, of course, we have to compete with big pharmas, which we are now here. However, for such kind of huge R&D expenses, newcomer is very difficult to catch up and compete with them. So that is the reason why. So what kind of space is the most workable for us? So that is regenerative medicine field, which is actually we are front runner right now. For instance, JTEC has actually 2 PMDA approved products in Japan, tissue products. And then we have actually materials, which is scaffold materials. Because of film businesses, film business has a very nano-level control of materials required. So then we had a very strong technologies for that controlling of functional materials. So that is another asset of ours. And then those 2 plus iPS cell, we can create actually really new important field of, for instance, organ actually design or the 3D assay. Those kind of actually new value can be created in the market. That's the technology strategy fit for us. The next technology fit is, I don't explain in detail, but simply say Fujifilm's material expertise and the knowledge technologies, and also the high production volume process technology engineering. Those kind of things are very great synergies between Fuji and CDI. So that is the current agenda for our company, how to create the synergies. Then CDI has actually biotechnology know-how and expertise, and Fuji has the processing engineering or the optic engineerings, and also machinery engineerings. Those kind of combinations can create the next generation of the production facilities. And we believe these cell businesses, production system could be the next very important competitive edge in the world. So then maybe last slide, this is actually introduced for the discussions for hours. We believe, however, still very challenging market. The reason is, this is very famous actually, innovation dilemma chart. We have actually still using primary cells or animal actually studies, and then the regulatory people are looking for those data. And of course, pharmaceutical people are also looking at those data. And then, because of the past data, then people can judge, so this is right or not. So then, in order to go to the next technologies, so some data accumulation could be very important, but that takes time, of course. So then, we think now we are IPS business in this shadow part. So then how to make a good gear for the next actually starting point. That's our challenge right now. However, if actually we can overcome that phase, then we can actually make a great growth as everybody sees next actually high growth of the stem cell businesses. And then, as I told, production could be very important for the next competitive edge. The reason is now almost the research phase has been ended. So then next stage is industrializations. Industrialization is always production capability is a key because the cost of a sense of cost, or the actually a sense of qualities and the high volume, or those kind of things are of course very important things for the management. And especially this IPS cell, we see scale out and scale up production necessity, because some cell can need a lot of volumes. For instance, if we go to the cardiovascular therapy, an enormous amount of cells are necessary. But then, for instance, eye treatment, so not so much cell is not necessary. So then also autologous and allogenic approach. We have to manage both, especially the scale out, how to manage scale out is very challenging for us. And then I listened to yesterday's manufacturing seminar in here, but then maybe same discussion was held over there. And then I think that's a very much challenge. But if overcome this one, then that company can get more profit, I believe. Then finally, maybe this is discussion point. We believe common goal among stakeholders is very important to overcome those early stage of business. I just listed up actually patients, advocate groups, and researchers, doctors, industry, founders, FDA. But those actually, of course, stakeholders have a different interest, so then sometimes actually conflict each other. Now, however, in order to speed up and then to make something, we believe actually some common goals of each, and then make a collaboration to break through the current situation, which is very important. Then I hope that will be
great.
Thank you.
Thank you. So I think that was a great overview of the state of the market, and I thought it was really interesting when I looked at that on what was the attraction for Fuji into this, because I know when I first heard about this acquisition, I kind of scratched my head and said, what's a film and camera company buying a IPS cell company? So I think this cleared that up for me. Asutosh, I wonder if you might comment. There were three challenges that Kaz laid out. One was around sort of this inflection point that we're at, I think, going from primary cells to IPS, and There's potentially other new technologies. There was one around the scale-up of manufacturing, and then this final one around stakeholders. Do you see others? Do you think these are legitimate challenges for the space we're in?
Yeah. Well, thank you very much. It was very interesting. And like you, I was scratching my head when I first heard about it. But when you look at the strategic overlap and the kind of decline of one business and looking to where you diversify, it kind of makes sense. So in terms of the challenges, I think, you mentioned 3. I think there is a number of others. So I think when you look at the regulatory challenges and how they differ across different jurisdictions, I think that's one. There's also something about lack of standardization that we have. We really don't have any common measures that can be globally referred to when we talk about even how to count the number of cells. There is accessibility of the tissues and cells. So how do you have accessibility? How do people manufacturing? I read somewhere some statistic like there's about 320 trials going on at the moment. If even 20% of those go ahead and are approved, you have roughly 60 odd, production capabilities that are needed, and we just don't have the production capabilities to be able to manufacture these. Something you pointed out at the right quality. That's one thing. There's the clinical aspect of it. People who are able to do the clinical trials and are trained in this. The CGMP facilities. And then I think the scale-up investment, although investment is happening, people are still investing with a lot of trepidation and IP. So I think these are the number of things that we see.
Great. Just, we started talking last night a little bit about some of the differences globally, and maybe you could each comment on this, and you touched on this in your remarks, but it is not one world as you think about this today, and would you both be curious what you both think about that as you think about this market?
You mean different countries?
Yeah.
I see. Yes, I think we have the US and Europe and Japan. Those 3 regions are very important regions for this business. And then especially research purposes, I believe Europe and also USA is a little bit ahead compared to Japan. But now Japan is actually, very much speeded up to catch up those kind of things. So then probably more or less same situations will be created in near futures. And then, but clinical part, of course, Japan is very much ahead.
Yes.
Because of the regulatory change.
Right. Certainly one of the more innovative approaches to regulatory we've seen, yes.
Then I hope FDA will change mind.
I don't know, those of you who are in the plenary session, I know what, I think we're still a ways from there, yeah.
Yes.
Yeah. Asutosh, do you?
Yeah, I think I'd agree with what you say. There is a difference between the research and the clinical, especially if you look at the number of papers that are being published. But when you look at clinical applications and also investment, there is a difference in the 3 markets. I see, I guess from a regulatory perspective, let me take that first. Japan is miles ahead. When you look at the numbers, somewhere between 200 to 400 billion by 2050 is this market. I think Japan looked at this and said, we have got to do something. We want a piece of that. And they looked at this as having the potential to really change lives significantly. So they've just gone ahead and done it. There are, of course, risks, but we're seeing already investment flowing to Japan from the US and from Europe. And we're already seeing approval of therapies there. Europe, I see as almost a little bit of a halfway house between the 2. They have hospital exemptions and other ways of doing it. And of course, you have the complication of 29 member states or 28 member states. But they have a lot of good research that's going on, and they also have clinical advancement. And the US has a lot of great research and great potential, but the regulatory system is perhaps still, in a sense, using the drug approach as opposed to looking at it from a different paradigm, which is cells are not drugs.
Right.
And I think the question is, how do we enable that? I think the challenges are also slightly different in that the environment in the US is still, depending on what you want to do, stem cells still have this, kind of stigma attached to it,
Yes.
much more than we see in Europe and much more than we see in Japan. And I think that stigma is going to be key to overcome, depending on where we see the market going.
And we started talking last night in particular around embryonic.
Yeah, so embryonic is a great example. In the UK, the first application went in from the Francis Crick Institute for research involving, basic research involving gene editing using using CRISPR-Cas9 for human embryos. And that's just for basic research, it's not for clinical applications. But if that's approved by the the authority there, it'll be the first regulatory approval of gene editing. So that's, and I could not imagine at the moment, given the environment that we have here in the US, that being approved, even if it's just for basic research.
Yeah.
And there was the first clinical application and treatment of a young girl at Great Ormond Street Hospital using TALEN technologies to treat her for ALL, so lymphoblastic leukemia, and she's been cancer-free for a year. So there is a huge difference I see in terms of at least perception there.
Yep. In the previous session, I sat in for the first 40 minutes or so before we met, and there was some discussion about cost and the cost of these and the cost of cells. And I mean, would either of you care to comment? I mean, is this going to ultimately save money for the system in your view, or is it jury still out? I mean, where are we on it? In theory, this, the dream is, of course, you have diabetes, I give you a cell treatment, and you're cured for the rest of your life, and you live to be 110, and die crossing the street because a bus hits you or something. So, any thoughts on that?
Well, I think pricing is very difficult at this moment because, for instance, such kind of chronic disease, normally lifetime cost of that treatment of disease are very huge.
Yep,
right?
very
But
expensive.
if actually some cellular therapy is effective at once, how to pay those kind of lifetime costs at once, or how to divide,
Right.
those kind of things are very difficult.
It's particularly complicated in the United States where we have a private payer system as opposed to
There may be some insurance ideas is necessary. And also, because of these kind of new things, always R&D cost is very much incurred beforehand.
Right.
So that means from the industrial company point of view, so how to actually pay back those kind of investment as early as possible, and then actually stabilize prices. Then those kind of actually consideration is very important. How to make some consensus with the market. That's, I think, a very big challenge.
Great. Do you have any comments?
I think cost is one of these things that's going to be so difficult. And I think that's for a number of reasons. One of the arguments is this is not palliative, this is cure. So how do you put a price on cure versus a lifetime worth of treatment using injections or pills? I think there are a number of strains of thought that I see coming out. Some people have the argument that this is going to cost a few $1,000s as opposed to hundreds of thousands of dollars. And then pricing comes out and they find out the treatment is actually $100,000. And that shocks everyone and they kind of go, whoa, this was supposed to be cheaper. But I think the issue there is that is a one-off as opposed to $100,000 over 10 years, which ends up being $1 million. So that's one aspect. But I think the other aspect of the economics of this that I think perhaps sometimes gets missed is it depends on whether it's auto versus allo. And what I mean by that is it's a very different game if you're in the allo market potentially, because allo really looks at a product. So the whole economics of that is based around having products, which is what pharma has always done, which is how do we manufacture this large scale to sell it to millions of people in a bottle or as injections or whatever. Auto is personalized medicine. And for that, there are two different challenges. One is, how do you get investment, and therefore which affects the ultimate cost?
Right.
So how do you encourage investment into that area? Because given the current regulatory system and the challenges that we've described, people don't want to invest in this. So you don't even know that there are things going to be available. They don't want to invest in this, which of course affects the pricing because they can't see it coming up, because it's personalized medicine, because the patient target group could be a few thousand, or even 50 if it's small children.
It becomes a very orphan situation.
For a very orphan situation. That's the first thing. And the second thing is, that could be changed if we see some kind of Japanese approval system where people have approval post safety and efficacy, and some kind of reimbursement, which changes the economic model.
Right.
So I think that's another thing that affects the pricing, which is auto versus allo.
Terrific, thank you. So I've done lots of talking and asked all the questions. What I'd like to do, we've got about 10 minutes left. If there are any questions from the floor, we'd be happy to entertain them. And we've been, come on, there's got to be one. Because there's people I know in the room I'll cold call, so. Sean. Mm-hmm.
All that, with mixed results. I'm wondering what the panel's thoughts are about how we're going to actually grow out these cells on an industrial scale, with that understanding that the regulatory authorities talks of a xeno product is probably not the best.
As far as I know, still regulatory considerations has not been clear yet for us. And then we are trying to figure out what kind of actually real requirement will be in the future. And then probably regulatory side itself is very much actually struggling to find out what is the right way to keep the safety. But then same thing actually, we are also exploring together with them, so what is the real safety is. So then it's a little bit harder to say, so what kind of directions it should be at this moment.
Sean, I could envision three potential outcomes. We either solve the serum-free problem, which there's lots and lots of people and there's lots and lots of effort going on. So that would be, I think, the optimum outcome from everybody's standpoint. There's sort of at the other end of the extreme is the regulators convince themselves that even though there's a xeno element to the process, if you will, we can convince ourselves that we can get it safe enough, and that in particular where you've got orphan conditions that there is no alternative, one could envision some acceptance on the part of regulators, certainly in some countries, to accept a xeno-based medium. Or there may be some middle ground where some have one answer and some have the other. Long term, my hope and thought would be that it's going to be the first one, that we'll crack the code on the serum-free, but we're probably a ways away from that. I know we're working on that right now in my company, and it's not a simple thing. I mean, it really isn't. Do you have any?
Yeah, I mean, I think the xeno serum-free issue is a great point you raise, and that kind of goes to the thing that we were talking about, manufacturing challenges, and absolutely. And I think we will probably see one of two things happen. You'll either see a tidal wave of evidence and pressure mounting up to require regulators to look at this in a different way, meaning that we have to have a different way of proving safety on that. So what are the different markers or endpoints that would have to prove that? Or I think we'll see a disruptive technology. something which will be completely left field, which will come in and is going to change that. So we'll see us moving away from the xenoserum-free. One of the two. I think I foresee one of those two.
So when you, to a time frame to a specific milestone, is that?
Time frame to scale, time frame to, scale is a big one in terms of the ability to manufacture.
Sort of
Time frame to therapeutics, and obviously that's
a mature industry, multiple therapies, we can produce it on a reasonable basis. Yeah, okay.
perspective on that. How are you thinking about it as corporate CEOs?
I see.
You can answer it first. The consultant, we'll let the consultant answer it first. How about that? Yeah.
Okay, so maybe this is really personal view. We are now engaged in the iPS cell-related businesses. And in terms of iPS cell, so then probably early IND case is 2017, what I said. And then maybe 2 or 3 actually cases with IND will be 2018 or 19. So then if actually US does not change any regulations, 10 years is necessary to actually go to the market. That means 10 years after, so those therapy actually huge businesses is taking off. Then, however, research tool part, I believe drug discovery part, those disease diversified models can be started to be used in the big pharmas. Maybe so you saw our newsletters which is related to the deal with Roche. So then we expect actually pharmaceutical companies are trying to use IPS for their assays to screen the drugs. So that means those volumes is increasing maybe within the 1 or 2 years range. And also other aspect of toxicology testing part, which has been already a lot of discussion was made, and then it is ready to go. So then once regulation has been, for example, ICH regulation has been changed, so then all actually toxicology assay is changing to the, for instance, stem cell usage part. So that take off very quickly. So that's my rough view for the futures.
It's really how long is a piece of string sometimes. I think it depends on a couple of critical things, and one is so maturity depends on which part you're talking to. Full maturity of the market, whether it's just research, whether it's clinical. I think the research aspect will see maturity much quicker, the basic research. We'll see advances in that, like we've seen in the last 18 months, 2 years, and it'll really start to fly more and more. That's inevitable. I think the maturity of the applications, the investment, the manufacturing, the larger investments that are needed is really dependent on the policy and regulatory environment. Because whether we like it or not, this comes back to investment. The environment depends on how comfortable do people feel putting their money behind something which they don't know if it's going to be allowed, right? So if I don't know something's going to be allowed, I'm not going to go out and get money to fund it. And in that uncertainty, the maturity will take longer. So I'm hesitant to give you a figure. Is it going to be 10 years? Is it going to be 5 years? If the FDA and the US government changes the legislation or regulatory environment next year, I think we will see a huge surge in investment, which will lead to things that we don't even envisage. But I think that is really a key turning point, because it brings to the market a lot of stability, a lot of certainty, which perhaps people are hesitant to. So there's a direct relationship between the investment and the maturity.
So yeah, I just want to echo, I think barring a change in the regulatory environment, we're probably 10 to 20 years away from any kind of industry that's scalable. My own view, and this is just my own opinion, is we can't afford that. I mean, if you look at what the United States spends on healthcare, if you look at the demographics in every major country in the world, I believe the regulatory environment is going to change because it's going to have to, because we just cannot afford the system we've got. So I think with that, you could accelerate that. Maybe is it 5 years to 15? Maybe that's what it looks like, and you know, but who knows? Right? We're out of time. I want to thank everybody for coming. I hope this was at least an interesting and a thought-provoking session on a Saturday morning. And I really want to thank all of you for coming. I want to thank the group for the questions. And we hope you enjoy the rest of the conference. Thank you.
Thank you.
Thank you very much.